My name is Elanor Murphy and I'm in 8th grade. The most exciting thing about my project was that I was helping protect people. Since wind turbines are a new and helpful technology, most people are focusing on trying to make them more productive. My project, however, was focused on whether they could have any negative effects. Wind turbines are still an exciting new technology, but it is important to find out whether they could be harmful. Last autumn, I learned that many researchers are trying to discover whether wind turbines could have any harmful effects on people and animals nearby. These wind turbines are a source for renewable and alternative energy. There are lots of them all around the world, some near people's homes. The question is, how do they affect the people living nearby? One possible effect could be stress, caused by the low-frequency noise they emit. During this presentation, I will shorten low-frequency noise to LFN. But who was I going to test it on, though? I decided to use Drosophila Melanogaster, more commonly known as the fruit fly, as my test subject. Trying to keep these pests alive felt kind of wrong, but they are a very useful model organism. After exposing them to the LFN, I needed to be able to measure their stress levels. There are many possibilities, but the method I decided on was anesthetizing them and timing their recovery rate. The longer it takes for them to revive, the more stressed they are. The question I was asking was, does the amount of low-frequency noise that wind turbines give off affect Drosophila Melanogaster's stress levels? If the LFN emitted from wind turbines does cause stress, then it could be affecting both people and animals. The purpose of this project was to find out whether that was true. The first step in my procedure was to obtain and culture the flies. I practiced anesthetizing and transferring the flies a few times before attempting the experiment. It was trickier than I thought to keep them alive. But I did find that it was easier to count the flies when they were in a large clear box. The next part of my procedure was to cause them stress by exposing the flies to LFN. In this part, I left the flies in separate rooms with similar lighting and temperature and subjected the test flies to the LFN for 24 hours. The final section of my procedure consisted of measuring how stressed they were. The first step was to anesthetize them and observe how quickly they recovered. I noted the first fly to revive in each box, and every 10 minutes I counted how many were revived. Some of the data had to be discarded, though, because someone kicked the boxes while passing by. My results did seem to show that there were negative effects. At the beginning, the control flies were obviously reviving faster, as you can see by looking at the blue line on the chart. But as the experiment progressed, the test flies seemed to start reviving faster, as you can see by the red line, around the 1 hour and 15-minute mark. This, however, was due to the fact that some of the control flies died. I do not know whether this was from the accident or maybe some other reason. So the increase was most likely due to the fact that there were more test flies reviving. The information from the first hour is the most important and reliable because it shows the first reaction immediately after the anesthetization. However, more conclusive tests should be done to confirm this conclusion. Some variations I might try if I do the experiment again would be 1. Using carbon monoxide to anesthetize the flies instead of freezing, or 2. Determining their stress levels by measuring hormones, or 3. Measuring expression levels of longevity genes. The first change, using carbon monoxide, would be better than freezing because it would reduce how much I had to handle the flies. This would probably help reduce the number of flies dying during the experiment. I'm especially interested in the second and third possibilities because I realized during the experiment that when I was counting the number of flies revived, I was really counting the number of flies that were moving. If There were some flies that were revived but not moving. I was not including them. If, however, I was able to measure stress by looking at hormones and expression levels of longevity genes, then I would not need to worry about whether the flies were revived or not. This method would be much more objective. It is extremely important that more tests like this one be done, because if the LFN from wind turbines does have detrimental effects, then we would want to know as soon as possible. We would also want to investigate a solution that would still produce renewable energy, but would not be harmful to humans. Thank you for listening to my presentation.